So hello
Mhm.
all, I'm Craig Maddox. I'm CTO here at Moberg Analytics, and welcome to, I believe this is the 18th of our case study in Neurocritical Care AI webinar series. And if you told me we were going to have 18 fantastic, amazing presentations a year and a half or two ago, I would have, not sure I would have believed, but it's been fantastic. And today is no exception. Um, so, really as we say every week, the goal with these webinars is really to help to help everyone who's out there working on these issues and these problems in the mission of treating and understanding brain injury. And so, that we do that through a lot of different avenues here at Moberg Analytics, in addition to, being a commercial enterprise, of course. We do tutorials and webinars. We have starter grants to help people get started, where we provide tools and cloud time and consulting to get projects off the ground. So really, our mission as a company is to help all of you, and we do that through a number of ways. And these webinars are one of the ways we do that. And we get amazing people on every week. It's been so fantastic. Um, so going on, also please provide us feedback. We're always looking for feedback, suggested improvements, suggested topics. Please volunteer to speak. There are so many people out there in our audience who are doing amazing work, and we really love it when people step forward and say, "Hi, I'd like to share that." And we have things get posted to our YouTube channel and our social media, and we get, really great participation and engagement through all of those channels. So we really love being able to do this. If you have any ideas, you can reach out to that bit.ly link or email me directly, craig@moberganalytics.com. Also, we've got quite a nice library of replays built up. You can reach them through our YouTube channel or at the link at the bottom of the screen there. If you have questions, please use the Q&A feature of Zoom. Everyone's real familiar with that now. Also, just to let you know that we are recording this, and the replay will be made available via our website and YouTube channel and other social media channels. A new resource I'd like to announce today, or I'll let Ethan announce it. We started a neuromonitoring analytics Discord server, and this is something that Ethan and the team spun up, and we're already getting great engagement. And so I will kick it over to him and let him tell you a little bit about that.
Thanks, Greg. Hi, everybody. We created the Discord server as a resource to support the community investigating neural monitoring and analytics. It's open to everyone, and we hope that it's the start of a collaborative environment in which we can all work together to solve these really hard but exciting problems. So you can access the Discord server by the URL that's on the screen, neuromonitoring.com/Discord.
Cool. Thanks, Ethan. So today we welcome Shu Han from the University of Cincinnati. Shu holds dual BS degrees in mechanical engineering, one from Cincinnati and another from Chongqing University in China. He also stayed then at Cincinnati, got his MS in mechanical engineering, and is staying there again now, where he's working on his Ph.D. His research is focusing on machine and deep learning techniques to address data labeling challenges. And he's been working on, those are obviously a huge issue in the area of neurodata. And so he's been working on some cool stuff that he will share today about that. So today, he's going to present a novel methodology for subpeak identification in ICP waveforms. And then he's going to show us how that is used in the extraction and analysis of ICP morphological features. So with that, I will be quiet and turn it over to Shu and let him take it from here.
Okay, thank you, Greg, and thank you, Ethan. So let me share my screen to get started. Can you all see the screen just fine?
Yes.
Okay, all right, thank you. I'm very glad to have this opportunity to share my work here. My name is Shu Han, and this is the title of the talk is a novel methodology for intracranial pressure subpeak identification. And it's mentored by Dr. Brenda Foreman and Dr. Jia. And also, I would like to acknowledge also some of our peer students, Varun, Marcella, Alex, for their effort. So in this presentation, I will first give you guys a quick introduction to our research center and also the background of this research problem. And next, I will introduce the proposed methodology for the ICP subpeak identification that was introduced in one of our previous publication. And next, I'll present the results and also mention some of the follow-up work we've been doing recently on this same research topic, but it's an improvement based upon the method. And in the end, I will briefly mention a couple of future work and current work we've been doing. So first of all, our lab is called Center for Intelligent Metrology and Sensing, and Dr. Xiaodong Jia is our PI. The goal of our lab is to develop advanced sensing and monitoring solutions and technologies, leveraging the state-of-art data science and machine learning algorithms to solve the specific problems in the biomedical and manufacturing area. And we have worked on like a couple of biomedical applications, such as the ICP waveform analysis, which is what I'm focusing on, and also some other topics like the intravitreal drug delivery or the running-related injury. So for the interested audience, you can go to our website for more information on our research. So now back to the topic, the traumatic brain injury. So the traumatic brain injury is a type of alteration to the brain structure that is caused by blunt force or penetrating force that it can result in severe physiological and cognitive dysfunction and even worse results. So patients with traumatic brain injury may be recommended for intracranial pressure monitoring, the ICP monitoring. So the ICP basically measures the pressure inside of the skull, and it serves as a biomarker for the secondary brain injury. So normally constraining the ICP within a relatively normal range, like 10 to 20, could lead to like a positive patient's outcome. But fail to do so could also result in the ICP hypertension, which could lead to inflammation or brain edema that can cause the secondary brain injury. So current ICP monitoring are still like mainly focused on the simple mean ICP amplitude to guide the intervention, and this is often reactive. And as opposed to it, for the proactive ICP monitoring, we can analyze the ICP waveform to predict the episodes of ICP hypertension and such to guide the doctor for taking actions. So the ICP morphology, it's referred to as the ICP signal over one cardiac cycle. And the morphology of the ICP waveform can actually provide some insights to the patient's physiological state. And a normal ICP waveform consists of three main subpeaks, the P1, P2, and P3 here. And normally they are in a decreasing order in the amplitude. So the P1 is normally just dominating over P2, but sometimes the ratio of P1, P2 can change. And this could be used as a indicator for patients' cerebral spinal compliance. So when the P2's amplitudes start to rise up and even rise above P1, this could mean that the patient is experiencing a decreasing brain compliance. So identifying the locations of the subpeaks and quantifying their magnitude could give us opportunity to extract the morphological features such as the P2P1 ratio and even more for guiding the doctor and also indicating the patient's physiological state. However, from a data science perspective, it's not like it's a non-trivial problem, mainly because of these challenges. And the first one being the variation in the ICP morphology over time. So we have single peak, single subpeak, or two subpeaks or three subpeaks. So the number of subpeaks can vary, and also the morphology itself can evolve over time. And also, even for some waveforms with similar morphology, there could also be different relatively magnitude within these different subpeaks. So for this one, we have the P1 greater than P3 greater than P2, and for the second example, we have like them in the decreasing order. And the third one is that there could be signal noise in the real-time data and also missing data and also the irregular ICP waveforms. So there are a couple of notable existing approaches handling these subpeaks identification. There are the deep learning methods that was published in 2022 that identifies the P1 and P2. And also there are the famous mocap algorithms, and also there are the Bayesian-based ICP subpeak identification. So while all these methods trying to solve this subpeak identification, but they all like exist, there are still some limitations to be addressed. So the previous implementation are more or less mathematically complicated, kind of limits the ability to validate on the large data set. And also the performance of some of these algorithms is relatively poor in identifying the P2 and P3, which means that they could have a good accuracy on identifying the P1, but it's less accurate or not as good in identifying P2 and P3, which could not give us like the full set of morphological features. And also these works don't often incorporate the SME knowledge, the doctor's knowledge, to adapt the new subpeak waveforms or morphologies. And also the incorporating ICP, ABP, and ECG for multimodal analysis and cross-signal morphological features are not presented in these works. So for our work, our objective is to first keep the model simple, and also we hope to improve the performance in identifying these subpeaks. And also we want the simultaneous ability to address the ICP, the ABP, and ECG. And also we also want to like leverage the expert knowledge there to help us interpret the morphology and also make like a more accurate labeling. So now let's jump into the methods. And before I start, I just want to give you a quick heads up. The whole flowchart will look complicated. However, we split them into three main blocks, with each of them has a very distinct goal. So I will first illustrate the big pictures, then I'll dive into the details later. So the first part, or the first block, is about data preparation. So to begin with, we only have the raw signal of ICP, the ABP, and ECG. These are the continuous recording from the patients. So it's necessary to do some data pre-processing and also some initial labeling. And the goal of the second part, the second block, is the morphological classification. So the main idea of this is that we want to propagate the subpeak labels among the similar ICP waveforms. And the similarity is actually the key to our method. So here we want to do a morphological classification. So we train a SVM model to do that. And the third part of the method is called template generation and subpeak identification. So this is where all the subpeaks of new data and incoming data are identified. A key method we will use here is the dynamic time warping, which I will talk about it later. So the data we use here are the ICP, the ABP, and ECG. They are from the patients with severe traumatic brain injury and admitted at UC Medical Center. They all have the comprehensive neuromonitoring that allows us to get these data. So the ICP signal was measured by a ICP probes that were placed under 2.5 to 3 centimeters below the inner table of the skull. And also the multimodal data was captured by Moberg's bedside monitoring. So we have continuous recordings for 9 hours for each of these signals. And we have 9 hours for each of the patients for a total of 10 patients. And those data will be investigated here. So let's talk about the detailed method. In these methodology slides, I will put a block here, normally on the upper right or left, to indicate the goal of the current step, so that you can keep track of what we're doing here. So the first step is the data segmentation. Here we have the ECG, the ABP, and ICP signals. And the first step is to do a R-peak detection. We use some simple transformation on the ECG signal and also a peak finding algorithms to find those R peaks so that it indicates where each cardiac cycles are. Then according to those, we can identify the onset points for ABP and ICP for all the continuous recordings. So here we break them into each individual waveforms. And this is what we end up getting. So the label shows the ICP local max, the onset points, and eventually each individual peaks is our focus in the later analysis. And the second part is the second step is the outlier detection. So in these signals, it actually contains outliers, noise. So we want that to be removed. So those can be caused by patient actions like coughing or reading errors, instrumentation errors, and such. So the way we're doing this is by extract the summary statistics for the waveforms. So for each of the ICP, ABP, and ECG waveforms, we extract some summary statistics and set thresholds for each of the statistics, and eventually we can filter them out. So this is what we end up getting. Like the outliers are shown in the red lines, and still we kept the labels for local max or the onset points. So now, as I mentioned to begin with, we only have the raw signal. There is no label at all. but what we want to do is the subpeak identification. So it's necessary to do this initial labeling such that we can continue. So we have data from 10 patients and 9 hours of recording from each of them. So we split them into 30 mini batch for each of the patient. And in each of the mini batch, it contains these set of ICP, ABP, and ECG waveforms for 1,000 waveforms. Then we will select the select the mini batches and to manually label some of the waveforms. And also with those limited labels and the labeled waveforms, we will train an SVM, the support vector machine waveform classifier. So at the end of this step, we will have this SVM model, which is the morphological classifier, trained on very limited manual labeled data, because there are a lot of it's intensive labeling work there. So, which we'll call this like a base model. It will do the job, but it will do the job very poorly in terms of the morphological classification. But at least it can make some predictions. So the goal of the next step is to iteratively improve this classifier so it can do a better and better job. And here we use a framework called semi-supervised learning. It's essentially using both unlabeled data and labeled data to train this machine learning model, and which is exactly what we have here. We have very limited labeled data, but largely unlabeled data. So for each patient, we have 10 mini batches. We will use 10 mini batches of these unlabeled waveforms. So all of the data here are unlabeled and unseen by the SVM model, but we will feed them into the SVM model anyways, and to let the SVM generate a prediction. So what the SVM generates is a thing called posterior probability, which is basically a measure of how confident the model is. And if the posterior probability is low, it means that the prediction is bad. So that could be because of the unseen waveform. So whenever this happens, we will pass it on to the subject matter expertise, the doctors, to assign the labels manually. That includes the morphological labels and also the subpeak labels. And we do this iteratively. So after assigning that label, we will pass that back to that SVM model. So we do this iteratively to refine this model. We do have a comprehensive diagram to guide us about the stopping criteria, but I will not go into the details of that. But essentially, at the end of this step, there are two things we can get. The first one is an improved or updated SVM model, the classifier. And the second one, we can also have a bunch of labeled training data, which includes the manual labeled ones and also the model labeled ones, but with confidence. So with all that, the next step is called template generation. And here we use a technique called dynamic time warping barycenter averaging. It's basically a averaging algorithms that is based on an algorithm called DTW. So DTW stands for dynamic time warping. Since it's very critical in our work, I'll just spend a minute to talk about it briefly. So I took these dummy plots from a DTW tutorial, but it shows what DTW does pretty well. So essentially, for example, we have two time series. They both have these key, we call it features, but they are not aligned in time. So with a normal Euclidean distance, or normally we will just pair them one by one. But what DTW does is that it will help you align these key features at each discrete time points, so that you can generate, the DTW can generate these type of alignment pairs. Then the DTW distance is just calculating the Euclidean distance, but instead of calculating them by street pairs, we calculate them along the warped path, so that we can know how distant, how far away are these two time series when we exclude the time difference, because they are now aligned. And this is actually a key to our later subpeak identification as well. And back to this DBA thing, the DBA basically leverages this DTW distance such that it can align a bunch of individual sequence and make a centroid out of it through this iterative process. And eventually what you can get is a representative waveform of a group of similar ICP waveforms. It's centroid, it's an averaging not only for the magnitude, but also for the time. Because it in this way, in the DTW way, we can compensate that time difference there. So at the end of this template generation, we end up having a set of template libraries, which are labeled by the subject matter expertise, and also from the iterative semi-supervised learning. So here, the criteria for selecting the waveform template are based on the number of subpeaks and their relative amplitudes, and also we will consider the overall ICP waveform morphology. So here we have subpeaks template with 3 subpeaks, 2 subpeaks, and 1 subpeaks, and with also each of the subpeak have like a different relative magnitude. So basically, this includes the variation, most of the variation in from the 10 patients data we selected. So as of now, we already have a template library, and also we have our well-trained SVM morphological classifier. And the next step is to propagate that subpeak labels. And here we use a technique called the derivative DTW, the DDTW, which is a variant of dynamic time warping, but it keeps that similar core ideas. So the dynamic time warping, the derivative dynamic time warping basically takes the first derivative as information to generate those warping paths. And for it, because the ICP morphology, essentially, it's the derivative, the information is contained in that first derivative. So in this case, it's actually more robust than a traditional DTW because of that similar shape. So this is what we can do, is to align these subpeaks such that, so the red lines is basically the subpeak mapping that's generated from the DDTW. And by aligning the subpeaks of incoming samples with its template, we can actually know where are those subpeaks are for different morphology labels. So one note for that is this method works regardless of the number of subpeaks. So no matter it's 2 subpeaks, 1 subpeaks, or 3 subpeaks, it just aligns that similar feature being the subpeaks there for between the incoming samples and the template. So we can also project this onto a continuous signal with the subpeaks label and also the onset labels. So that's basically the subpeak identification part. Then from there, we can extract the so-called cross morphological features because as long as we know where those subpeaks are, we can start to extract the ICP or cross-signal morphological features. So we've got a table of these features between ICP, ABP, and ECG, which contains the amplitude or the latency between these subpeaks. And here we show that one example from one patient on a continuous segment of signals that the P2, P1, P3, P1, P2, P3 are extracted. And also in this segment, you can clearly see that there's a nice trend of the P2 decreasing, which probably indicates that it's getting better because in general, we assume that the P2, P1 ratio, the larger the P2, P1 ratio, the better in the brain compliance is. So now with this method, we also want to evaluate quantitatively how good this method is. So the way we do this is through 3 independent reviewers. So for each of the reviewer, we will randomly select 1,000 subpeaks, 1,000 waveforms from each of the 10 patients. Then 3 independent reviewer gives us a total evaluation of 30,000 waveforms. And when the subpeak labels assigned by the algorithms is plus or minus 5 data points to the ground truth subpeaks, we will call it a successful or accurate identification. So the table shows our detailed quantitative metrics from each of the reviewer for all the 10 patients. So from this table, you can see that the total accuracy is 98%, 98% for the subpeak identification. And also one thing to note is that for each of the patient, the algorithms can perform consistently well. The lowest is probably 94%, but other than that, it's pretty consistent and robust for the method to identify the subpeaks. And we also benchmark our method with some other existing methods we mentioned. And our methods holds a couple of advantages. So the first one being that it can accommodate the ICP, ABP, and ECG simultaneously for that cross-morphological feature analysis. And our method is also evaluated or benchmarked extensively on 30,000 waveforms from 10 patients. And the proposed approach can also can identify the P1, P2, and P3 when one of the peak or 2 of the subpeaks may or may not exist. So that's also holds as an advantage. So up to this point, we basically wrap up the previous proposed methodology, but we're still keep working on some of the improvements or limitations. So here it's the proposed 3-block diagram we just mentioned. So from this method, we can actually get some abilities that can be thought as pros of this method. which are the data pre-processing capability from the raw data. And also we have validated this DDTW-based Subpeak identification as a robust algorithms to do the Subpeak labels. But it also has a weak side, which is the block 2, being that for this semi-supervised training framework, we will need constant involvement of doctors to adapt unseen waveforms, which is kind of labor intensive. So we want to come up with a way to improve that. And also, we have noticed in some of other publications that there could be a different perspective on these interpretation of the ICP waveforms. One specific being that with the advance of AI technologies, the deep learning, there are models or works that tries to directly do a classification on these ICP waveform morphologies. So basically, there are, in convention of these references, there are 4 to 5 types of waveforms, being the normal, the possibly pathological, the likely pathological, and the pathological. So this means when patients is being assigned in each of the label, that still in reflects the P1, P2, and their relative amplitude in some sense, but in a different way that make them into distinct classes. So in our follow-up work, which we already summarized them into like a paper, and it's under review, our objective is to improve based upon these observations and limitations of the previous method. So the first one is that we want to reduce the human labeling effort, and also, but in the same time, we still want to keep the morphological interpretability, because we think having the SME knowledge and make them interpretable is one of our advantage in the previous method. So we still want to keep that interpretability. But in the same time, we also want to consider like the classification aspects of things, because it's essentially discussing the same thing, the morphology, but in 2 different perspectives, the classification and the subpeaks. So the third one is that we also want to have this real-time monitoring ability, because all of these biomarkers like P2 P1 ratio, if we want to make it in real time, so that the method can detect any subtle change regardless of classification, or from the subpeaks perspective, we want to detect the subtle change in this morphology change. So here, as an improvement of our aforementioned work, we first enlarged the data set significantly to include a 1.2 millions of waveform from 60 patients. So here, I showed a scatter plot in 3 dimension, which we obtained from some dimension reduction algorithms, just for visualization of these ICP morphology distribution. And later on, we process this whole data points, the scatter points, with an algorithm called mapper from the topological data analysis to construct a mapper graph, which is basically a bunch of nodes connected by the edges. So here it contains 456 representative ICP waveform templates. So the template used here, or we constructed here, is basically analogous to the template library we extracted from the DBA in the previous work. But it includes a more comprehensive variations of that ICP morphology. And here we label them in class 1, class 2, class 3, class 4, and the class 5 being the artifacts. So for these classes, we adopted the conventional class assignments represent these class 1 to 5. But for each of these nodes on the graph, it actually consists of a group of similar morphologies or ICP waveforms. And the blue line is the centroid, is so-called the centroid of the template. And by having this, we can color this whole graph with the class. And you can actually see that from the class 1, it transitions gradually to class 2, class 3, which means that that P1 is decreasing, or say the P2 is increasing, so that it transitions from class 1 to class 4. Eventually, it contains only one subpeak there. And also with this graph node, we actually have a bunch of different ways that we can color the graph. We still holds the same structure there with the same node, but instead of coloring them with the classes, we can also show some morphological features on this plot. For example, here we use this P2 over P1 ratio as an example. So from the same view angle, you can see with that waveform classes transition from 1 to 4, there is actually a decreasing in these color bars that shows the P2, P1 ratio is increasing, which is consistent of what we define here. The P2, P1 ratio is increasing, and the P1 amplitude is decreasing. So here we used in this process, we still kept the expert knowledge here, being that the 456 nodes, we will still have SMEs manually label them and examine the morphologies. But we actually significantly reduce the labeling burden from million level to just a couple of hundreds. And also, this is like a one-time effort. It's not like continuously involved in the process, but this is a one-time effort, so that for the later samples, for the later incoming morphologies, we will just match the similar nodes to the incoming samples, so that we can do this subpeak identification in a similar way as mentioned previously. Excuse me. And also we can project different morphological features as shown here, the P2P1 ratio, to show the different patients' physiological status. So with that graph we have there, we actually want to apply it or adopt it to some real-time applications. So here we have the ICP waveforms, the raw signals from one patient, and with the same way we can extract the P2 and P1 ratio there. And in this time window of 30 minutes, we can actually see that the P2, P1 ratio is increasing. So that indicates during this 30 minutes time window, the waveform, the morphology of the waveform is transitioning. So with our method, we can actually project each of these individual waveforms, which onto this pre-generated graph that I showed before and show their status in real time. So for this 30 minutes time window, we split them into, so the algorithms split them into 6 distinct window, which in each of the window, it has this different morphology for that ICP waveform. It's changing from time to time. So before, we can only blindly see or even just extract this feature and knows that the P2P1 ratio is changing, and that is all. But right now we can actually split them into these type of windows, and having each of the window being the distinct or slightly different ICP morphology. So in each of the node, we can also see that the P2P1 ratio is indeed increasing, suggesting the same thing. So the scatter plot here is actually the same graph generated from the million level data set, but here the idle nodes, the nodes that are not matched, being not similar to the incoming sample, are noted as the gray dots there. So here, it's basically another view of looking at this transition or a new way of visualizing this ICP morphology transitioning. And we think that for each of these underlying dots, the reason that there's a reason for the waveform to be staying in the same node. So we're hoping that this proposed method could potentially be used to monitor that change in the patient's physiological state, and also keep track of some key features such as the P2P1 and even more over time, and visualize that in a way that in these trajectory on the map. So eventually, we're hoping that by using this type of visualization and looking at the trajectory, we can sort of know, combining the patient's status right now, we can sort of know where this patient is going. So that's the ultimate goal for predicting that patient status deterioration. So for these up to now, we basically wrap up the existing work we've done, and there are a couple of future work that we're working on right now. So the first one is that we want to actually make this whole module or functions into a neural network because of the real-time implementation speed in a clinical setting. But different with training a neural network or deep learning model from scratch, we have this already existing morphology map that we know how to interpret it. So with that, we can always come back and say, okay, the deep learning model is probably not doing a good job, and we want to retrain it, and we know what we're doing right here. So the second one is that we want to test the actual clinical utilities for these ICP morphological features we just mentioned, and evaluating if the trajectory is indeed leading to some clinical outcome. And the third one is that we also want to validate the ICP morphology on a even larger variety of patients' data and variety of ICP waveforms to ensure, and also to ensure the performance of the algorithms. And last but not least is that we are working with Moberg Analytics to try to create these type of modularized analytic tools that can be implemented on the Moberg's platforms. So eventually we will hope that this can goes into the bedside monitor or go have some like real usefulness in the practice. So that will wrap up this talk, and I will take any questions from now. Thank you.
Well, thank you very much. That was an amazing talk. And we're really excited to see that last bullet on that last page. I think some of the most exciting things that we do are are working with researchers to, take this amazing work of years and years and years and study and bring it back around and bring it back around and put it in the clinic, put it at the bedside. So we're really excited to be able to hopefully do that. There's a number of people working on really cool things, and getting them back to the bedside is one of our reasons for being around. So thank you. Can you see the chat? There was a question in the chat. This was a really interesting talk. I haven't heard of dynamic time warping before. Why did you decide to use dynamic time warping instead of normalizing the time axis for each waveform? So each waveform lasts for 1 arbitrary units, for example.
Okay. Good question. Let me go back to the slides where we're talking about this. So here I'm using like a dummy plot, but the reason that we want to align them is actually we think that there are informations in the time axis as well, because that reflect onto the feature being the latency here. So from the onset points, the latency to like maybe P1, P1, P2, P3 could mean differently. So we don't want to, because if we normalize that, we will basically lose that bit of information and treat them as all aligned at the same time point. But we also want to consider that bit of information so that we can sort of consider it in an implicit way. And also for using dynamic time warping, it's also quite distinct to think in that it just naturally aligns these subpeaks. That's also one of the advantage of using the dynamic time warping here. So it's kind of by its nature, we can do like averaging the DBA, the dynamic averaging, standard averaging with DTW, and we can also take one step ahead to align them, the subpeaks. I hope that answers the question.
I thought I saw a hand up somewhere.
Hello.
Roland?
Hi, I'm Roland. I'm one of the PGY2 residents at Emergency Medicine at Cornell Columbia. Great talk. I was just curious, do you have any code that's like available to search experiment the derivative of like DTW and DTW itself in like a code base that's like currently on like GitHub for sort of our sort of novice researchers like myself?
Yeah, so the DTW is actually a tool that has been there for a long time. So this is not a new thing. What we do here is that just to take this tool. So if you're asking for the the DTW, there are like a bunch of tools in MATLAB or Python, depending on what platform or what coding language you're using. But also in our method, we also plan to sometime open source part of the code, but it's implemented in MATLAB. We're still working on just transitioning the whole thing into like a more implementation ready code in the Python. But right now we only have the MATLAB version of that. We can probably share that on GitHub link if you're interested to study that.
Great. And just another question. I saw you. It was I don't know if I correctly understood, but it sounds like part of the effort that you're describing in the first part is essentially trying to identify like the subpeaks with like the SVM approach. versus like the
Mhm.
distinct classes. I'm not sure if I understand this correctly, but is there any utility in sort of having an agnostic view of like the waveform and looking at the waveform overall rather than sort of just trying to distinguish each individual peak and looking at the ratios to predict like IC peak prices or like rising ICP?
Yeah, so in this first part of the work, we are more just trying to designate this the waveform labels based on what we see from these 10 patients. So basically, we have eventually 10 labeled a 10 template here labeled by SMEs with their distinct subpeaks, but this basically covers a lot of the variations that coming from these 10 patients. So it could shift a little bit, but essentially by averaging them, we will have this type of waveforms. So so to your question, no, we will just basically uh, generate this templates from what we see from the 10 patients. But later on in the second part, we actually make some improvements to make that agnostic so that we assume that the whole data set here contain different variations or as many variations as possible that there could have. So in the ICP morphology. So in that sense, we don't have to have a expert like checking this if this ICP waveform or how should this be labeled. We just have them all and having them clustered by this method so that we will have a quite comprehensive variation of waveforms, if that makes sense.
Gotcha. Yeah, great. Thank you. That answered my question. Sorry, just like a last quick question. And in terms of, have you looked at like the decay kinetics of these like waveforms? If there's any data that's encoded with regards to, like the rate of rise and like the decay of like the ICP waveform that specifically correlates or provides any additional information with regards to like ICP, or is that's not something that you were interested in?
Yeah, so in the scope of this work, we're more onto just the morphological analysis. But I think that is indeed like some of the future work that we can do is to explore what this graph means. Because right now, basically, we are having this graph. It's underlying like connected by this waveform morphology. But what does it really means? And is there any really significant use in clinical setting by reading these where like the patients are in these nodes, we can sort of know where the patients will go in terms of the physiological status. So that's part of the future work. Yes.
Got it. Great. Thank you. Great talk.
Thank you.
Cool. I don't see any more questions in the Q&A. I'll give a last call as we're about 1 minute from the top of the hour. Okay, great. Shu, thank you so much for coming on and sharing. Oh, wait. Rohit had one. Can you see the question, Shu, in the chat, or do you want me to read that?
Oh yes, I think I can. So, are there any features you're exploring beating the templates? Let me first go back to the slides. I think you're asking the morphological features, right? Well, no, this one. Yeah, so when building these templates, we're not looking at specific morphological features like P1, P2, although we do consider them like implicitly, being that just they show as different waveforms. And of course, they will have this different P1, P2 ratio or other morphological features underlying. But when constructing this template library, we just do it based on what we see, if that makes sense. And if you can like ask follow up if I didn't answer your question or I didn't understand your question correctly. But I'm going to the second one regarding the real-time implementation. Current approach not feasible. Oh yeah. So the second question I think is asking the kind of the implementation speed or complexity. So we have tested like we have timed this algorithms to see how fast it can goes, and based on our previous observation, I think it's still pretty fast, and it's roughly like it can process the waveform like 100 waveforms in 27 or some 30 seconds. So it should still meet the real-time implementation needs, but the network is just easier to be like implemented or deployed in terms of the hardwares, because like neural networks are networks. Once the network is trained, it doesn't matter like what type of device, like computation device you have there, or what the Python environment you need. It's just neural networks, so it will have a lot of pros in terms of the real deployment.
Cool. Well, Shu, thank you again so much. We look forward to following you in this work, and we look forward to implementing it. So
Yeah.
it's really, really cool stuff. So thanks all
Thank you.
for,
Thank you.
yeah,
Thank
so
you for having me here. Yeah.
thank you. Yeah. And for all the attendees all across the United States and beyond, thank you very much for showing up on a Friday afternoon or evening. And the recordings will be available on our website and YouTube. So thanks very much, and we'll close the call.
Thank you. Bye-bye.